Welcome back. So today we will discuss about the structure details of the muscle fiber. So in the last class we discussed about the myofibrillar structure, and then what are the sarcolemma and what are the components in the individual muscle, starting from skeletal muscle, visceral muscle, and then in cardiac muscle. Now we will try to look into the detail structure aspect in terms of anatomical model. What we call anatomy is the actual internal aspect of the muscle fiber. So if we look into the muscle fiber as such, we have this long myofibril, and in myofibril we have this kind of striations. So these striations are equally spaced, and we will see in detail why these striations are basically are causing this muscle fiber to look differently. Now, the major thing here we need to understand is this muscle fiber is kind of not in a single entity, right? These are having a combination of all those protein molecule. Now we will discuss this one is darkly colored, and these are made up of myosin filament, and these are made up of actin filament. We will see it later. Now the actual thing is we have multiple nucleus here. So the nucleus is not a single one. So the nucleus is distributed throughout this striated muscle fiber entirely length. Now in biology, always there is some terminology associated with this nucleus related stuff. Nucleus is, of course, we are using karyo. Karyo stands for nucleus. So we can always call anything related to karyo is nucleus. Karyology is the science which deals with the study of nucleus. Again, similarly, prokaryote means anything which is primitive. Pro means primitive, karyo means nucleus. And similarly, we have eukaryote, which is true nucleus. U means true. Now here, since we have nucleus in more in number, we are having this multinucleated condition. So we have this multinucleated. So we have multinucleus. We can say multinucleus. Now this multinucleus condition is called synthesia. Synthesia is a very specific terminology used in biology in order to show or in order to depict any cell which is having multinucleated condition. So more nucleus is called synthesia, and this is nothing but it's a multinucleus condition. This dark and light bands are basically reflecting, behaving differently with the light. So this dark area is called anisotropic. Now anisotropic stands for non-uniformity towards the biophysical parameters. Non-uniformity towards the biophysical parameter in different direction. So this is a uniform this is a terminology which is used to depict an area which is not uniformly showing biophysical characteristic, while we have another area which is light in color, and we call this light area as isotropic. And this isotropic is something which is uniformly showing a pattern towards the biophysical parameter in all direction. So isotropic is a very specific terminology for uniformity, and as anisotropic is a very uniform terminology for non-uniformity or ununiform structures. So in short, these are called as A and these are called I bands. A and I band stands for anisotropic and isotropic. Now since we have this idea that these are very distinct two banding pattern, we can nicely look into. So we are discussing about this myofibril, what is happening in case of this muscle fiber. In between this I band, we have now before going in detail, let's see what are the actual composition of this one. Since this is an area which is anisotropic, these are made up of myosin. So this is made up of myosin, while this isotropic area is made up of actin. And in the last class we discussed that actin are thin filaments and the myosin are thick filament. The diameter is little higher, more in as compared to actin. Now in between this I band we have a distinct line in between. This line is called Z band, or simply it is called Z line, that is alphabetically last letter Z. So you have here also a band line, here also you have line, here also you have line. So in between isotropic band we have a line in between, and this line is called Z line. Now in between this Z line and the area, the length in between two Z line is very specifically called as sarcomere. So this length in between two Z line is called sarcomere, and this is an ultimate unit of muscle contraction. So basically at the time of, we will go in detail about this whole phenomena, what is happening in terms of physiological aspect, we will discuss it later. But this is the unit length. Once we are getting a contraction kind of phenomena, this whole sarcomere is kind of squeezing, whole sarcomere is contracting. Now because of this contraction we are having some movement in the muscle. So let us see some other marker here. So we call it sarcomere. Now so we have like in between anisotropic band, anisotropic area which is made up of myosin protein, we have some lighter area which are called as hinge. So these are hinge area. In short, these are called H. So we have hinge, we call it zone. So we have hinge zone in between this anisotropic area, and in between hinge zone we have a specific line called M line. So in between this we have M line. M line. So we have hinge area, we call it H, we have hinge area, we call it H. So in nutshell what is happening, we have complete picture of myofibril, and what we are looking here is the area which are darkly stained, and darkly stained area is called anisotropic area made up of myosin, and in this dark area we have a light shaded area which is called the hinge region or hinge zone, and in this hinge zone, in between hinge zone, in the center we have a line called M line, and after this area we have a light shaded area which called as isotropic area, and this isotropic area is made up of actin protein, and this actin protein containing this area is having a line in the straight, and this line is called Z line, that is the last letter of the alphabet. Now this is in total. Now what is happening here, that this structure is quite uniformly seen in this muscle fiber. Now what we will see here now, so we will just try to simplify this one by deleting all, and then we will just mark the area here. So this is M, this zone is H, this is Z, Z, and then we have Z. Now we will see this structure, what is there in this Z area. So in between these two band, in between these two Z band, the area is called sarcomere. Now we will try to look this area very carefully in detail, that what is the actual mechanism, this is very grossly looking picture, but now we will go and see what is there inside this structure. Now if we see this Z line, so we will draw this Z line in a different style. So we have this Z line, And then from the Z line we have this. So now this area, since we are saying it isotropic area, and these are made up of actin. So these are the fiber which is basically nothing but the actin filament. And similarly from this Z we have another Z here. So we will just try to draw a similar kind of a structure here. So we have another line, and this is also the actin area. So basically what we are trying to look into, that we will just try to see that what is there in this area. So this is Z band, and this is Z band, and this is actin filament, and in between actin filament we have this area which is anisotropic, and this anisotropic area is what we call it myosin. Now this myosin is, so this is just looking like floating, right, and this is not correct. So what is happening here, this myosin is kind of attached to a very spring like structure with the Z band, just to give a flexibility to this myosin filament. So this area which is giving the attachment of this myosin protein with this Z band is called titin. So this titin is kind of springy material, spring in nature, which is calling this one to be flexibly coming and going out at the time of contraction and relaxation respectively. Now what we have seen, that this particular area is showing little bit of the lace stain. So this is the hinge region, and in between hinge we have this area, we called it M line. So we have Z line and we have N line, and this whole Z to Z is what we discussed earlier, is called sarcomere. So this sarcomere is nothing but the distance in between two Z band or Z line. Now in normal process what is happening, this Z band is kind of contracting, and because of this contraction and relaxation we are getting muscle movement, which we will see in the actual mechanism of this whole protein. Now we will see the actual composition of and how these are made up of. So we will see what is actin and what is myosin in detail. So we have to understand that what is the protein content, what are the actual ingredient which is available in actin, making this one as a very special structure, and here also causing this one as a very specialized structure as a myosin. Now as we know that protein is nothing but the polymer of amino acid. So if we add lot of individual amino acid, we can form a protein. So similarly this actin and myosin is nothing but protein, but the character of these two protein are entirely different. So we will discuss this one in again detail. So let's start with the actin first, and then we will discuss about the myosin. So in actin, let us start actin. Now this actin is available in two forms. One is called globular actin and the other is called filamentous actin. So this is nothing but like this globular. So we can simply call as globular actin. Now this globular actin is nothing but a very spherical kind of entity. Now if we add lot of globular form of actin, we will make a filament. So basically if we add lot of globular actin, we will make an F actin, which is nothing but a filamentous form of actin, which is filamentous form of actin. So we have two form of actin, one is called globular actin, G actin, or F actin, which is filamentous form of actin. Now so what we have is an actin filament. So basically this is the actual unit. In every globular form of actin, we have a very specifically designated area which is kind of active site. So, this we can simply say it is an active site. We will see the actual use of this active site later. Next is we have another filamentous structure which is running in parallel to this actin molecule, and this is called tropomyosin. Tropomyosin. So, this is kind of running. So, there are two filamentous structure running parallelly to this actin filament, and these are called tropomyosin. So, this is tropomyosin filamentous structure. Apart from this actin and tropomyosin, we have another protein associated with this actin filament, and that protein is available in triad. So, we have this kind of triad protein distributed throughout the length of this actin filament, and this triad protein is called troponin. So, troponin, tropomyosin and actin, these are the three composition of actin filament. Now, this troponin, which is available in triad form, is not the single entity, we are calling it a tetrad. So, this one is either I or T or C. So, there are three. So, one is I, the other is T and the other is C. So, I troponin, T troponin and C troponin. Now, this ITC stands for the area where this troponin molecule is adhering or interacting. So, this troponin, I troponin is interacting with the actual actin protein. So, these are kind of interacting with the actin. The name is coming from isotropic nature. So, isotropic, we already discussed about the isotropic nature, that the isotropic is coming from actin. So, this troponin I is interacting with the actin filament, actin protein. Then is T. The T troponin is interacting with tropomyosin. So, this is tropomyosin. So, this is interacting with tropomyosin, and the last one C troponin is interacting with calcium. So, this is calcium ion. Now, we will discuss about this calcium in detail in few minutes, that these are very important ions which are responsible and acts in the contraction mechanism, because calcium is very important. In order to do contraction, the sarcoplasmic reticulum or the content, the muscle has to release the calcium ion into the area where their contraction is going on, because calcium is a kind of initiator. We will see the whole phenomena in detail. So, just summarizing this whole actin protein, actin filament. So, this is an actin filament made up of globular actin. This globular actin is having an active site in between. We will see this active site because this is interacting with the myosin later on, and this globular actin is polymerizing to form a filamentous form of actin, and this filamentous form of actin is having another parallel running filament called tropomyosin, and there is another protein associated with this whole structure, and which is called troponin, and this troponin is having three flavor I, T and C, and respectively these are interacting with actin, tropomyosin and calcium, or these are modulated, or their activity is very much related to the presence of actin, tropomyosin and calcium. So, this is about actin. Next we will see about myosin. So, let us see that what is there in the myosin. Now, actin is thin and myosin is thick filament. Now, in myosin, this myosin is a very different kind of protein as compared to actin. Now, this is kind of a structure, these are helical structure, and this is a head kind of region, and this is another head. So, they have two heads, and these are the two head region, and then this is we can simply call it this as a tail and this one as a head. Now, this head region is and the tail region, these two are segregated into different structure in this myosin. So, we are calling this one as meromyosin. So, this is head meromyosin and this is tail meromyosin. Sometime this meromyosin, so these are two units are distributed. This is meromyosin, this is also meromyosin, but this is called heavy meromyosin. So, the terminology is heavy and this one is called light. So, light meromyosin. So, there are two distinct area in this myosin protein. One is called head, which is globular in nature, and these are called heavy meromyosin. Then we have a tail region, which is called light meromyosin. Now, these are all helical structure. The tail is the helical structure. Now, very uniquely about this structure, we will discuss this structure here. So, if we see this head region, what is there in this head region is this globular kind of structure. There are two sides here. One side is interacting with actin protein. We have discussed in the last actin structure that each actin globular form of actin is having this active site. Now, this active site is what is interacting with this portion, this area in this head region of myosin, and the second one is interacting with a protein, a kind of a very important, not a protein, actually it is a very important aspect in muscle movement or any energy dependent process. We will discuss it now in next 15 minutes or so. Regarding this structure, this is interacting with energy currency of the cell, and which is called adenosine triphosphate. So, this is an area which is having ATPase activity. So, what is happening? ATP is binding to this area and then ATP is getting hydrolysis. So, the process is called hydrolysis, and because of this hydrolysis reason or hydrolysis process, the ATP is converted into adenosine diphosphate. So, ATP is converted into adenosine diphosphate plus phosphate, and this released phosphate is what is giving energy for this contraction process. So, the whole process is under the control of, requires some kind of energy, and this energy is coming from this adenosine triphosphate, which is binding to the head of this heavy meromyosin, which is in the top of this myosin filament. So, coming to this whole summarization, this is myosin filament distributed into light meromyosin, which is tail region, and this is head region, which is heavy meromyosin, and this head region is having two sites. One is binding to the active site of globular actin and the other is binding to ATP molecule, and this ATP is providing energy at the time of contraction. So, now we will discuss about the mechanism, the actual mechanism of muscle contraction. So, let us start mechanism. So, mechanism of muscle contraction. Before going to this molecular aspect of or the actual detail of the mechanism of the muscle contraction, we have to understand some basic stuff related to this contraction phenomena. And one very basic is this whole process is kind of requiring energy. So, in body processes, in human physiology, there are two distinct processes. One is called passive process and the other is called active process. So, there are many processes going on inside our body every day, and day to day basis our body is doing these two processes only. So, either the processes are active or the processes are passive. Passive stands for there is no consumption of energy. So, no energy. So, energy is not involved in these processes. whereas in active kind of processes we have energy dependency. So, energy is required to do these processes, active processes, and this energy is coming in the form of adenosine triphosphate, which is simply called as ATP. So, ATP is nothing but the currency of energy which is required in order to do an active process. And what we are discussing now is the contraction, mechanism of contraction of muscle is an active process because it requires adenosine triphosphate in the form of energy. So, we have to understand that energy dependent processes are all active process, while energy independent processes are passive processes. So, these are the two normal process. So, we will stick to this active process as such here in the mechanism of muscle contraction. Now, these are two processes, and there is another aspect in this whole contraction, and what we will do, we will try to link this whole thing. Now, what is happening? So, having understood this one, let us see another one. Now, what is happening? Suppose we are having an insect bite. So, what is happening? We are getting an insect bite. So, once we are getting an insect bite, the insect after biting is doing something, and from that part we are getting a response towards central nervous system. So, in central nervous system we are getting an information that look, something is doing some kind of activity in the hand region, and immediately the brain is giving some response to the hand in order to do some activity, either to pull the hand or to do some sort of activity. So, there are two aspects, either we are getting an information in the first instance, and the second we are doing some response. So, if we try to see the aspect that what is happening, suppose this is central nervous system, either brain or spinal cord. The central nervous system is comprises of brain and spinal cord, and then we have muscle, which we are discussing here. So, we have muscle and then we have central nervous system. So, there the connectivity between the muscle and the central nervous system is happening because of a very unique kind of cell, and that unique cell is called neurons. So, neurons are the cell. Now, what is happening? We are getting, so suppose there is an insect bite here. So, we are getting insect bite in the muscle. So, muscle is getting information and then transferring this information to central nervous system. Now, this kind of neuron which is sensing the outside kind of response. So, we can simply call this one as periphery response, and this is central nervous system. So, anything which is coming from periphery to central nervous system is called sensory. So, these are sensory neurons which is giving an information, and after receiving to central nervous system is immediately giving some response in the form of something that is maybe contraction of muscle, which we will discuss now, to muscle, and this response is called motor. So, this is motor neurons which is giving information to muscle to do contraction or do any other phenomena. So, just to summarize this phenomena or this aspect, because we are going to see the mechanism of muscle contraction, and for that reason we need to understand this one. So, anything which is coming from periphery to central nervous system is called sensory neurons or sensory phenomena, and something which is coming from central nervous system to muscle is called motor. Now, what we are getting an ultimate response is contraction. So, contraction is the one aspect which we are working. So, we need to study this area, right. So, what we need to understand is how this motor neurons is going and giving this information or this kind of impulse to muscle, and for that reason how the muscle is starting contraction. So, let us see what is happening in this area. Let us draw again. So, this is central nervous system and this is the muscle area, and then we are having this motor and this is sensory. Now, we would like to know more about this area. So, let us see. So, this is a neuron. So, this neuron, we will draw this neuron as the structure like this. So, this is the, we are not going to go and discuss about the complete neuronal structure. Of course, this is the body, and then we have this kind of dendrites, and then you have this one, this area is called axon. So, this axon is kind of supplying the information to muscle, and here we have our muscle. So, suppose this is our muscle. Now, what is happening? Once the information is coming from axon towards this muscle, this information is not coming in the form of direct contact. So, what is coming is a small chemicals. So, these chemicals are secreted in this area to transmit the impulse, transmit the information from here to muscle in order to do contraction here. Now, what is happening? These are the chemicals which are coming from this axon. This is the neuron. We will call it neuron. Now, this whole area is called synaptic cleft. So, this area is called synaptic cleft. This is presynaptic membrane because this is before synaptic cleft. So, these are called presynaptic, and this is called postsynaptic. So, this is postsynaptic and this is presynaptic. All right. So, here some information is coming in the form of some chemical, and these chemicals are very, very important in human physiology, and these chemicals are called nothing but neurotransmitters, and very well established example of neurotransmitter is acetylcholine. So, this acetylcholine is a chemical mediator that is transmitting the information from neuron to muscle. So, this neurotransmitter, so what is the name as such indicates neurotransmitter, the one which can transmit the information from neuron to some other part. Now, this area, this whole area is very important because this is interacting with this structure. So, this is a presynaptic cleft. Postsynaptic cleft, we have a chemical in the synaptic cleft. In some in neurology, where we are studying the neurons and all, this area is called synapse. So, this is synapse area where the two neurons are kind of transmitting the information with each other. Now, this should not be confused with a terminology called synapses. Synapses is exchange of chromosomal material, exchange of genetic information at the time of meiosis, and meiosis is a reductional cell division. So, we should not be confusing this whole terminology with synapses. Synapses is very important phenomena in cell division, where the chromosome, the structure in the genome, the structure in the chromosome are getting exchanged at the time of cell division. So, that is a part of meiosis. Here we are talking about neurons. The terminology is synapse. So, synapse is a very specific terminology where we are trying to see the exchange of information between neurons to the end. Now, since these are motor neurons, because we are discussing about motor, so this is called motor end plate. So, this is called motor end plates. So, this motor end plates are the ultimate area where this information is getting transmitted with the help of this neurotransmitter, and one such neurotransmitter, very well established neurotransmitter, is acetylcholine. Remember this one, that acetylcholine is one of the most commonly used neurotransmitter to transmit the information from neuron to motor end plate. Now, since the information is getting inside the synaptic cleft and then it is transmitted to postsynaptic membrane and then coming to this area, earlier the information was in neuron, now the information is transmitted to the muscle part. Now, what is happening here? The whole area is getting activated because of the impulse coming from the neuronal side. And what is happening here? The sarcoplasmic reticulum. The sarcoplasmic reticulum is endoplasmic reticulum like structure in muscle. So, in cell we call it endoplasmic reticulum. Let us clarify the whole thing and then we will discuss this one in little more detail. So, let us delete this part, it is not needed. So, we are just discussing this part. So, now what we are seeing here, that endoplasmic reticulum like structure in muscle. So, we have something called endoplasmic reticulum, we also called it ER. I am not going to discuss much about this ER here, because we have a similar kind of a structure available here in case of muscle, and we are not calling it endoplasmic reticulum, we are calling it sarcoplasmic reticulum. Now, sarcoplasmic reticulum is secreting calcium ion. So, calcium is kind of circulated or kind of getting out into the sarcoplasmic space. So, the sarcoplasm is the fluid content of the this muscle fiber, and the reticulum is called sarcoplasmic reticulum. So, sarcoplasmic reticulum is giving calcium out, and this calcium after coming into the area is activating the whole contraction process of the muscle, where the actin and myosin filament is myosin filaments are coming in close proximity and is starting the whole cascade of the contraction. Now, we will discuss about this contraction phenomena in great depth, because we need to understand that why this calcium and this information where the transmitting agent is getting transmitted to the muscle fiber, and how this calcium upregulation is causing the myosin and actin filament to slide over each other. Now, what is happening in I will just try to see that what else can be discussed here, just to simplify the whole story in small. So, we have this myosin filament, and then we have this actin filament, and then we have this band, we call it Z band, and then because of this contraction the muscle movement is happening. Now, this contraction is starting because of this calcium influx. So, calcium is a very important ionic, which we started in the discussion also. The calcium is very important ingredient coming out from the sarcoplasmic reticulum, which we have to remember that the calcium is coming from the sarcoplasmic reticulum and coming to the area space where you have a sarcoplasm, and this sarcoplasm is ultimately causing this contraction to happen. So, now we will see the actual contraction mechanism, and to understand this actual contraction mechanism, let us see the actual protein myosin and actin filaments. So, if we draw this kind of spherical structure, not spherical, it's a tail coiled structure, and then we have this unit, and we called it simply, there are two areas, right. One is binding to ATP, adenosine triphosphate, and other is interacting with, we will just slide here. So, this is an area that will be linking to, or that will be coming in contact with actin. So, we are drawing this one as myosin. Now, what is happening here, that once this myosin filament is interacting with the actin. So, let us draw this actin also. So, this is an active site of actin filament, and this active site is what is interacting to this actin binding site of this head region of meromycin. We called it heavy meromycin. So, this heavy meromycin is interacting with the actin, the active site of actin. Now, since here the binding of ATP is activated once the calcium is getting upregulated in the sarcoplasmic space, the ATP is binding to this area. Now, this ATP is getting dissociated into adenosine diphosphate. So, ATP is dissociated into adenosine diphosphate and phosphate. Now, what is happening here? So, there is a dissociation of ATP molecule, and this ATP will just mark it like a star. So, this is a dissociating ATP molecule. So, this function of myosin in order to utilize ATP and to convert this ADP and phosphate is called ATPase activity. So, ATPase activity. So, this should be very clear that ATPase activity is available in the head region of myosin. Very specifically, ATPase activity is located in the meromycin, the heavy meromycin area of this myosin filament, because this area is located. So, remember, ATPase activity is not available in actin filament, it is available in myosin filament, and that too in the globular head. Now, because of this reason, there is slight movement, and this slight movement is causing this whole head to move in this direction. And because of this movement, the whole thing is getting contracted. Now, because of this contraction, the muscle is getting squeezed. The muscle is coming close proximity. Now, if we start drawing the structure here, so we have this Z line, and then we have actin, and we have myosin filament here. Now, because of this contraction, so this is kind of sliding. So, this whole phenomena of sliding of myosin molecule over this actin is dragging this whole Z line very close to each other. So, sarcomere, which is the length of this Z line, is getting decreased at the time of contraction, and it may happen that this M will come very close to each other. So, this may be the possibility that this whole thing, so the earlier there was a space, but now there is no space, everything is squeezed because of this contraction. So, earlier the Z line was this, but now this is very small because of contraction. So, the sarcomere length is kind of contracting. Now, here we have getting contraction. This is called sliding theory of muscle contraction. So, this is sliding theory of muscle contraction. Now, here we have this Z line, and here we have Z line, and then we are having this filament actin, and then we have this actin is corresponding to this one. So, this is here, and this is the head of the myosin. So, this is causing contraction here. So, the things are coming in this direction. Now, here what is happening? Here also we have this. Now, here we are having this myosin head like this. So, this is also coming in this direction, and because of this contraction, the things are coming close to each other. So, because of this utilization of ATP, the things are contracting. Once the contraction is over, that means the ATP is completely hydrolyzed, right? We call another, I am just repeating, hydrolysis. So, hydrolysis of ATP is giving energy, and this energy is causing contraction. Once this contraction is over, the ATP is again it's adenosine diphosphate. So now during contraction you have full of ADP, this is converting again into adenosine triphosphate, ATP. So this ATP is again available for the next contraction cycle. So at the time of relaxation, the ATP is getting accumulated, and at the time of contraction, the ATP is utilized and converted into ADP. So at the time of relaxation, this is more, at the time of contraction, this is more. Now this whole process is energy dependent, as we discussed earlier. This is an active process. So anything which is so suppose if a person is doing very active exercise, very actively involved in doing muscle contraction and other kind of thing, there is a limitation of energy every time in your body. So body is having certain limited amount of energy during a time period. So if a person, suppose a gym worker, is always doing very rigorous exercise and all, energy is limited. So contraction is getting done and in the expense of that energy. After some times, when energy is gone, you will start getting a process which is called fatigue. Fatigue. Now fatigue is nothing but the tiring process, and this tiring process is coming because of the unavailability of ATP. So energy is going out, and if energy is not available, the body is not able to go further contraction or further kind of muscle movement, muscle contraction, and the process is called fatigue. This is simply called as muscle fatigue and all. We will discuss these two and other aspect in coming lectures, where we will try to discuss this fatigue phenomena with something else also.